So Gwen, Arun, it's so wonderful to be with you today. Thank you for taking the time to have this conversation. I'm really excited to talk about Cabaletta, what's going on with the company, your background, and then also Philadelphia and the ecosystem here. For those that are watching at home, I'm joined by Gwen Binder, who's the president of Science and Technology at Cabaletta Bio, and Arun Das, who's the chief business officer here at Cabaletta Bio as well. So before we jump into Cabaletta and the science, I'd love to start with both of you in terms of how you even got to this point, because there's so much richness in your backgrounds. Gwen, if we can start with you, you earned your PhD in viral immunology at Johns Hopkins. Then you went on to Penn here at University of Pennsylvania, where you worked directly with Carl June and Michael Malone on engineered T cells. And that include the original CD19 CAR T, which is like so amazing, went on to become Kymriah. I still remember when I saw the approval come through, I was like so, yes, exactly. Such a huge deal. So can you tell us, what was it like to be in that lab at that moment? And did you really understand at the time what you were on the verge of there?
Yeah, no, that's great. And I love to tell the story, so thanks for giving me an opportunity. And so it's actually, one thing I want to share with you and your listeners is that actually before CAR T in oncology, CAR T was developed in HIV originally. And so between Hopkins and Penn, I actually worked at a small company called Virexis that was founded by Bora Dropulic, and we worked with Carl. At that company, we're developing HIV gene therapy. And so there we developed a lot of the tools that ultimately led to the cancer therapies. So the lentiviral vector that's now widely used and is in Kymriah was originally proof of concept out of that company. So a lot of those early therapies failed, but they led to these tools that when I joined Penn, we worked on Z19 CAR T. It took 3 years actually to get through all of the preclinical manufacturing development work. NIH didn't fund it. We actually got foundational funding. You asked what was it like. I mean, it was pretty grueling. It was super long hours. We had to create everything from scratch. There's no regulatory roadmap. Nobody had done these things before. And all of the initial CAR T studies, even in oncology, had failed. They had led to very transient signatures. So when we went into that first patient infusion, we weren't really quite sure what would happen. And I have a personal story about that. I was actually invited to the first infusion. I thought I was too busy to go. I ended up going, and I'm trained, as a scientist. I'm not a clinician, and so this was my first time ever seeing a patient who was so ill and really only had a few months to live. You could really see it on his face. This patient's famous. It's Bill Ludwig. Now he's in the news and everything. And I was asked to explain to him what it took to get that study open, and I just felt kind of ridiculous actually talking through, these small steps in front of a person who was facing, the end of his life. And that patient ended up being cured and had a complete response and lived for 8 more years and ultimately died of other causes. And that, for me, was a really seminal moment in my career on several levels. One, I saw firsthand the transformational impact of this type of therapy that I've been working on for quite some time until then. And second, it really taught me patience. And if you have sound scientific principles and you work hard and you have a good team, especially it's all about the team, then you can actually achieve what you are setting out to do. You just never know exactly when it will happen.
Yeah.
So,
Oh my gosh, what an amazing, amazing story to see the first patient infused with CAR T. That's incredible. Oh my gosh. Can you tell us also, so you've taken that and you took that experience and you were at Adaptimmune and built up their basically whole US operation from, the clinical, the manufacturing, compliance, regulatory. What did it take to build something that complex from scratch? You're bringing so much experience to your work here at Cabaletta. Tell us more about that.
Well, I wouldn't recommend it. And not sure I knew what I was getting into. No, but it was a wonderful experience. When I was at Penn, Carl's group worked with a lot of industry organizations like Sangamo, Adaptimmune, and a few others. And so with Adaptimmune, when I was working with Penn, I opened 2 of their INDs, their MAGE IND, their NY-ESO IND. And so they said, hey, we want to set up a U.S. presence now that we have these INDs. Will you join us? And I said, yeah, sure. Let's see if I can do something else, you know. And At that time, all the manufacturing, the translational research, the clinical operations, everything was done at Penn. And to be a company that could actually develop the product into later phases of clinical development and be a standalone company that could partner and so forth, we had to set up independent operations, and we had to do it quickly. And so I said, oh, sure, we could do this in 6 months, 9 months. And you know, industry standards are different. And I learned a lot. You know, I hired people who had experience in the field. I hired a diversity of people from different companies, thinking that would be beneficial. So we had a lot of spirited discussions about the standards and so forth. But it's great, and I really learned that you know, to do something like that, you really have to have a fire in the belly. I had a real feeling that I could not fail. And the people I hired, the people I worked with had that fire in the belly and really did the crazy things I asked them to do, and we made it. We ultimately had 5 pharmaceutical companies come in and look at us, and they really passed us with flying colors, and it led to the GSK collaboration. So it was great. And so now at Cabaletta, it's fantastic because I get to do it again, but smarter. And a lot of the team that I had at Adaptimmune, you know, thankfully came along for the ride.
Oh my gosh, that's amazing, amazing. But I love that you shared that part of like, I wouldn't recommend it because it was like it sounds like there was like ups and downs, and that's the part I think people don't always get to see is like the challenge that it is to build something from the ground up, make those mistakes, but then learn from them and keep moving forward. So thank you for being so transparent and honest about that. And so for Arun, again, for you were part of Penn's very first class in the Vagelos program in life sciences and management, that dual degree track. We were actually just talking about it. It barely existed. So then from there, you went to Johns Hopkins Medical School. You were at Goldman Sachs for some time in investment banking and healthcare. Then you did a pediatrics residency at CHOP. So really cool path that you've taken to where you are now. And now you're the chief business officer. So what were you searching for with like all those different paths? And I'm not knocking it at all, because I think it's amazing when people do different things. So tell us a little bit about all of those experiences and what made you decide to take that path.
Yeah, I'm happy to. First of all, thank you for having me, Joanna. It's really a pleasure to be here. For me, I would say what informed my journey from the beginning, and my path ended up being different, but the journey at the outset was really driven by growing up having parents of friends who were physicians and realizing how many of them expressed the idea that they didn't understand why their administration, why their hospitals were making the decisions that they were making. So they were equipped to take care of patients on an individual basis, sort of case by case, but they didn't understand, why did the hospital not fund this? Why did the hospital shut this thing down or expand into this area or take over my practice? And for me, having an interest in medicine but never wanting to be a person who was engaged in a space where I didn't understand the context, I vowed early on that that's something I wanted to explore early on. So I wanted to gain the business context and the strategic context that healthcare entities operated in before going into medicine myself, with the idea that I always wanted to be a clinician, but not a clinician who didn't know what else was going on. And so that informed my decision to, when I saw the application for the Life Sciences and Management program, it was just, 1, a paper application, so that tells you how long ago it was. 2, it was just a checkbox, a paragraph and a checkbox of, do you want to apply to this program or not? And at the time, I had no idea it existed. It was the first year it was in existence, but I thought, this sounds pretty good. And thankfully, I did end up checking that box because I was fortunate enough to get in and had 4 wonderful years of learning interdisciplinary curriculum across life sciences and business. And it really prepared me, I think, for the winding path that I ended up taking. It was there that I knew I wanted to go to medical school, but before medical school, wanted to have some initial exposure into the world of healthcare and decision making. And so that's where Goldman Sachs came in, and I was fortunate enough to go there. In that period, applied to medical school, was admitted into Johns Hopkins, and had 4 of the best years of my life there. Absolutely loved it. I think they prioritize teaching you how to be the absolute best clinician you can possibly be. And for someone that had some interdisciplinary experience but really wanted to focus on being the best doctor I could be, that was the place for me. And so really enjoyed that opportunity. Took 2 years in between med school and residency to work in the consulting space, really helping to bring companies from academia into industry. One of those is actually what would become Cabaletta. And so it's the seed for what would end up taking me where I went back, was a resident at the Children's Hospital of Philadelphia. In that time, Cabaletta, as it was evolving, was turning into a company with a trajectory that was really completely unexpected. And while I was going through residency, maintaining connections with the scientific co-founders and the CEO of the company, realized that there was an opportunity to join a transformational, scientific, paradigm-shifting opportunity and a management team and a group that I had a deep level of trust in, and the opportunity to shape the culture and the team in a way that for someone at the stage of career that I was in was going to be unprecedented. For me, at least. I didn't see the opportunity to engage in such an effort maybe ever, and certainly not in the next 10 or 20 years. And so I always say I made the decision to leave clinical medicine, not for biotech, but for Cabaletta. And it's a decision I don't regret and have never regretted for the 7 years that I've been a part of the company. It's been a wonderful journey so far.
Oh my gosh, what a great story.
Thank you.
What a fabulous story. And like, I love that that's what influenced your decision of like, because to your point, like we as clinicians, we often want to practice medicine. We want to do the science. But we don't understand always the other side of things, like the business side and why decisions get made. So smart to like take that path. I really admire that. And you're kind of taking us into, sort of the next stage of Cabaletta. Like, as you said, you joined at the seed stage while you were doing your residency, and the science and obviously everything was drawing you to it. And I know, Gwen, you were on the scientific advisory board for a little while before you actually joined the company. For somebody who doesn't know anything about Cabaletta, how do you describe what you're doing here in very simple terms, so that, because one of the things I also am trying to do is help people who are not in the scientific community understand the science of what it is that's happening, because it's so cool, it's so cutting edge, it's so groundbreaking that especially in the autoimmune space. So can you share with us what is Cabaletta? What are you about, and what does it mean for CAR T therapy to be resetting the immune system?
So Cabaletta is solely focused on autoimmune disease and developing cellular therapies, autologous cellular therapies to develop durable cures for patients with autoimmune disease. When you think about what autoimmune disease is, in just plain language, like we all have white blood cells and they are responsible for protecting the body against foreign things. And your immune system is able to discern foreign from self through a process called central tolerance and peripheral tolerance. What happens with autoimmune disease is sometimes the immune system gets confused and it loses the ability to discern. Or maybe you have an infection and your body mistakenly sees the infection and your own tissue at the same time, for example. So you break that, what usually it's peripheral tolerance that you're breaking. And so then you have a subset of your white blood cells that are auto-reactive. And so where our type of therapy comes in is to basically target those auto-reactive cells and permanently deplete them from the body so that your body can maintain a state or regain a state of peripheral tolerance. And so that's a potentially curative approach, because you're basically getting at the root of disease in patients. And our original technology was a little bit more precise, targeting specifically a subset of B cells, a type of white blood cell that produces a pathogenic protein that causes the autoimmune disease. It's an antibody. Our current approach is a much broader approach that targets all B cells transiently, because we now know that B cells can drive autoimmune disease not just by producing pathogenic proteins, those antibodies, but also by driving a broader autoreactive immune response. They're basically the fuel for that autoimmune fire. So if you can get rid of all of those B cells transiently, your body produces new non-autoreactive B cells and you go into a state of health again. And so this is a durable, potentially curative therapy. It has been, curative. It's provided, 4 years ongoing clinical remission off therapy in patients so far. So
it's awesome. That's amazing.
What would you?
I wouldn't change it. Next question.
Yeah, I mean, the potential for the curative to cure the diseases, especially for this patient population, and we're going to get a little bit into that, is just incredible. Because the way that I think about it, just in kind of layman's terms, coming from an oncology background, and now is like you're basically giving somebody a whole new immune system. Essentially, the fact that you're able to do that and allow their immune system to stop attacking their body is just incredible. So I want to jump into some of the things that you've done to be really like strategic and thoughtful to make sure that this gets to patients as quickly as possible. And so you are running 5 simultaneous reset phase 1-2 trials as part of the reset program in lupus, myositis, systemic sclerosis, myasthenia gravis, and pemphigus vulgaris. And then your early data across more than 40 patients shows that the patients, they're coming off of all of their autoimmune therapy and not needing any therapy at all. So in some cases, so when you're looking at that data, what does it feel like? Like, what are you thinking about? Like, what's going through your mind when you see those curves or you see that data? And like, and even maybe, I don't know if like that patient that you were talking about, that first one that got CAR T, like what's going through your mind when you see that data? And I mean, really, when we say data, those patients, like...
Yeah, I mean, so you kind of hit the nail on the head. Like the dramatic responses we saw with those initial patients in oncology, it's very similar to the dramatic responses that we're seeing in autoimmune disease. And I think one thing that has surprised us, and I think Arun can talk more about this because he's done a huge amount of work on the different indications, but I think one thing that has surprised us and other people in the field is how broad the effect is, how many autoimmune diseases actually can be mitigated by targeting B cells. There is a history of effort there using rituximab, an antibody-mediated depletion, but it's not as good because it doesn't deplete from the tissues. And so I think what we're seeing here with CD19 CAR T is when you do deplete B cells from the tissues, which is where most of your B cells are and where most of the autoimmune disease is being driven from, when you do that, you're seeing effects even in diseases that weren't improved by rituximab. I think this is really exciting. I think there's a lot of science still to know. The mechanisms of resistance in autoimmune disease are different than in cancer, right? Autoimmune disease is a lot more tractable than cancer in terms of you're just targeting B cells. They don't mutate easily. They don't escape the CAR T easily. But there could be other mechanisms like long-lived plasma cells that might play a role. They're not targeted by CD19 CAR T, and this may be a form of resistance in a subset of diseases. And so there's no way to know this without actually treating the patients. And so we're really excited to see, not just the breadth of the indications we're going after, but other companies are going after in academia. So that, we all need to learn from each other and discover this together. And there's a huge patient population that can benefit from this. And maybe you could take it from there.
Yeah, I think the characteristic that resonates with me most strongly across all of the indications that we're assessing is the question we're asking with CAR T is totally different than with really any other drug that's been brought to market before. It is, you have moderate to severe active disease while you are on or have been on 2, 3, 4, 5, 6 medications. And what we're asking is, even though you have moderate to severe active disease with these medications, we're asking you to stop all of those. And then we're saying, take this one-time infusion and let's see what happens. We're going to keep you off all your medicines. And if you have disease come back, we can put you back on the medicines. But the goal of the study is actually to see how long you have meaningful disease response while staying off all your medicines. And so that's the thing I think that is hard to put into perspective on paper and with curves and graphs versus when you actually hear from patients and hear from physicians. If we thought about this study, anyone in the industry 4 or 5 years ago, people would have thought we were insane. Like, you're going to take my patient off 6 medicines that has severe disease or moderate disease and then give them a one-time infusion and see what happens? How? And yet what we're seeing is pretty reliably patients get deep responses fairly rapidly, and those responses are quite durable in the follow-up that we have for date. Now, Gwen brought up some great points around mechanisms of resistance in subsets of populations and other data that we and others are generating in the space. But the idea that the vast majority of patients are seeing these types of responses, it's amazing to be a part of. And it's something that no matter how many slides you put together, how many graphs you put together, hearing someone tell you that I don't have to take steroids, I don't have to take rituximab, I don't need IVIG, that to me is the thing that is just amazing about what CAR T is able to, the potential that it has realized so far.
Yeah. Oh my gosh. Like, and even just hearing you say that, like, you're going to stop 6 of your meds. Like, and I'm like, if I'm a patient and I hear that, I'm going to be like, what? Are you insane? So that is a lot of courage too for patients, not just their physicians, but like for the patients to trust that this is going to work for them. But now it sounds like you have like a lot under your belt already. So it's a little easier for patients to say yes, which is awesome. And that actually takes me to this particular patient population, because autoimmune disease is different than oncology. And this is actually something that I've been talking a lot about, because this is a population that is primarily focused or impacts women more than men, younger ages as well. They're healthier. I think you talk about also they often have commercial insurance, which we're going to kind of get into the reimbursement part of it. And they're working age, they might be of childbearing age. So there's a lot of factors that play into the type of therapy that they decide to take on as a result of their disease. And I know that one of your and I love this about the company, one of your top values is patients first. And I really believe, like, just seeing how you've evolved your clinical program and really thinking about the patient. Tell us about how you have thought about that, because this is a very different patient population too. Like I've always said, like, you can't just take the CAR T model of how we've been doing it in oncology and just like drop it into autoimmune disease. And I think a lot of people believe that too. So tell us about how you've been thinking about that and how you've maybe adjusted or designed your clinical trials to really meet the patients where they're at.
I think there were a couple of guiding principles for us that led to the way we thought about CAR T development in autoimmune disease. One was we had the privilege of access to the early academic data from Germany, where the group in Erlangen was already evaluating CD19 CAR T across a number of different indications. Those indications that they initially reported data on were lupus, myositis, and systemic sclerosis. And so if you see parallels between those indications and what our first indications were, it's because we followed the data. And the number one thing we wanted to understand is clinically, where is there proof of concept for CD19 CAR T and for B-cell depletion more broadly to enable a meaningful impact for patients. So that was number one. We leveraged as much about the conduct of that study, weight-based dosing, a construct that was similar, a manufacturing process that was similar, and essentially just brought that into industry-sponsored studies across a broad portfolio, because we felt like there are a lot of diseases where B cells are implicated in either the pathogenesis or maintenance of these diseases, and being able to achieve a deeper depletion than has ever been able to be achieved by any kind of small molecule or antibody-based therapy was worth evaluating. So that's why we started off with weight-based dosing, the same dose across a portfolio of studies, and then let that initial clinical data guide us. The other thing I'll say, and this is to your point on differences in the autoimmune population versus oncology, is we recognized that safety was going to be a key priority for this population. And so as we generated initial data with the construct with preconditioning, we started doing work to understand what are aspects of the product offering that may make a difference for patients. And I would say the biggest bit of feedback we received was, if there's an opportunity to eliminate preconditioning from the regimen, the fludarabine and cyclophosphamide that typically precedes CAR T therapy, that would be a huge market expanding opportunity, and patients would come to this therapy, especially in diseases like lupus, which does predominantly affect women of childbearing potential, to increase access to the therapy. Now, that is something where because of the experience we've had, and I think Gwen can probably talk to more, we recognize that as an opportunity. We worked early to incorporate no preconditioning arms into some of our studies, and that's data that we're actually really excited to be talking about later this year in the first half and in the second half. It all starts with the rationale for why we think no preconditioning could work, but sort of the foundation is making sure we're developing innovations that are patient-centered and optimize the patient and provider experience. So if the no preconditioning aspect is important to get into or helpful to get into more, Gwen, I think you can talk about that, but that's our sort of philosophy.
Yeah. The potential to eliminate the preconditioning is like huge on so many levels. So yeah, if you want to expand on that, I'd love to we can.
Just to riff on something that Arun was saying is that we, the patient-centric attitude that we have, today our lead indication, yes, it's using apheresis, it's using preconditioning, the manufacturing turnaround time, maybe around 6 weeks, it's a in-hospital infusion, there's preconditioning, but the vision is for the future, and we have data to support each of these steps, is in the future it will be a blood draw, a collection of cells, it will be a quick turnaround in manufacturing of 4 weeks or less, it will be no preconditioning, it will be an outpatient infusion, so that's just a single infusion. So then the patient experience becomes, when the patient goes in and they are prescribed Rese-cel, they give a blood draw, come back a few weeks later, get an infusion, and go home with some remote monitoring. And so we believe that's really possible. Right now, even in oncology, almost 50% of infusions are being done outpatient. Regarding the preconditioning, the scientific principle to us seemed very clear, because back, to my original comments, the original CAR T was done in HIV, and preconditioning was not used, and there was excellent engraftment and persistence in those cases. Preconditioning is required in oncology because not only does it make space or release homeostatic cytokines to basically effectively increase the infused dose of cells, because they expand more once they go into the body because it's a living drug, but the preconditioning also increases the tumor leakiness, the inflammatory status of the tumor, making it more immunogenic and easier to kill, not just with the infused CAR T cells, but also with the endogenous immune response. And it also reduces immunosuppressive cell types that inhibit an anti-tumor response. So those two last things aren't important in autoimmune disease. So we reasoned that if it's just a cell dose thing, we should be able to go without preconditioning and possibly just need to tweak the dose up a little bit. We published this at the ESGCT back in October. It's in our corporate deck. And so we started there with no preconditioning at the same weight-based dose as we're doing with preconditioning, and it's much better actually than we expected, where it looks like it's a threshold dose where some patients respond and some patients have the full B-cell clearance and look like we're responding, and there's one patient that doesn't have full B-cell clearance, is not responding. And so that tells us that we're close to where we need to be.
Yeah, it's amazing. I'd like to kind of shift now to just how you've been able to accomplish all of this in such a short period of time. You've really taken a very accelerated regulatory path. Tell us a little bit about that and how you've worked closely with the FDA to ensure that you were able to have a pretty accelerated path and then be able to then maybe expand into some of these spaces of the no preconditioning and so forth. So,
like I said, back when I was at Penn, there were no regulations, and even when we were first at Virexis developing the first lenti, we had to create everything from scratch. So I'm used to thinking about things logically, right? And so we as a team, we had actually talked about CD19 CAR T back in 2019 when the first animal model showing that it could work in lupus was published, and we considered it, but we were focused on our AA CAR platform at the time. So when that first patient was published by the Erlangen group, we said, let's go, because we already believed in the concept. So we were already ahead of the game. So it was only a year later that the first 5-patient cohort was published showing that, and then everybody said, oh, this is real, but we believed it before then. So further to what Arun said, we really copied what the Erlangen group did. We didn't get too creative about it because we knew it was good enough to stay with the 9-day manufacturing and to use a construct that was substantially similar to Kymriah, with one change, which is that we have a fully human binder, which then will be less immunogenic, right? And so then we were able to negotiate with the regulators, look, and we showed this and we published this data, look, our construct has the same potency, the same behavior as the Erlangen. Look, they're dosing at this dose and it's having this safety and efficacy profile. We don't need to do a dose escalation. Mm-hmm. And so we were able to very quickly go through phase 1 design where we say, okay, we're going to do 6 patients with each cohort. We're going to have independent INDs so that we can enroll in parallel, which speeds up clinical development and also separates out disease-specific safety signals were we to see them. Mm-hmm. So that's sort of the early phase strategy to moving quickly. And maybe Arun can talk about some of the innovation and how we were thinking about the pivotal study, given the dramatic treatment effect that we were seeing.
This effort was led by David Chang, our Chief Medical Officer, both in terms of the overarching clinical strategy and then specifically how we thought about the registrational studies. And there, I think he and the team that he's led really came upon what I thought was a fundamental difference. And it speaks to actually what we were talking about earlier, which is the major difference in what we're seeing with CAR T versus any other modality of therapy is patients are achieving the outcomes that they're achieving without any other supplementary therapy. And so if you look at rituximab, Safnello, Benlysta, any of these other biologics that have been approved in autoimmune disease, they are add-on to standard of care therapy. And so then when you're measuring the treatment effect in those scenarios, you likely will need much larger studies to detect a difference between treatment versus control. In Rese-cel's case, or with autologous CAR T, what we realized was if your endpoint is inclusive of the idea that you're achieving these outcomes without medicines, what is the likelihood that any other patient is going to achieve that same outcome? So a moderate improvement on the total improvement score in myositis, a major improvement on the total improvement score in myositis. Yeah. What is the chance that a patient who did not receive Rese-cel but also was taken off all of their medications would be able to achieve that at 16 weeks or at 52 weeks? And that was the driver for the efficient designs that we've been able to employ to safely develop this for patients. It's if you believe that the likelihood is really quite low, then the number of patients you need to treat in order to detect a difference between treatment and what you would expect is much lower than it would be in other studies. And so that was the biggest thing, is that our primary endpoint in the registrational study is not achievement of a certain total improvement score. It's achievement of a certain total improvement score while being off all immunomodulatory medicines. That's the big difference.
Yeah, that's huge.
Yeah.
Oh my goodness,
Yeah.
that's so huge.
Yeah.
And I'm just hearing, as I'm listening to the two of you talk about this, and I'm getting excited about it. I'm hearing all of the excitement around it. And what is it, I just am curious, you talked about David Chang and his team, to give us a peek behind the curtain, what is it like day to day working with your teams, to be doing this incredible science and the impact that you're having on patients? People must be just so jazzed to come to work every day. Yeah.
It's awesome. As you were talking, I'm literally getting goosebumps, not on purpose, but it is amazing, honestly. Yeah. And I feel so fortunate to be working with the group of folks that we get to work with at Cabaletta. We were just talking before, Joanna, there's a picture in the corner of this room from our IPO back in 2019. And I think one of the things that I'm most proud of about being at this company is that the group that came together in 2019, we've had one CEO, CFO, President of Science and Technology, Chief Scientific Officer, Chief Medical Officer, Chief Compliance Officer, Chief Business Officer. All of those people are the only people to have ever occupied their roles at Cabaletta, and the only person Cabaletta's ever had in that role is that person. So when you think about the camaraderie, the ups and downs, the building of relationships that we've all had over that time, it's so exciting to come into work and tackle new problems, new questions, new achievements with those folks around the table, and now with the teams that each of us have built. it's awesome. That's my first statement. I don't know what you would add, but I absolutely love it.
I mean, it's hard to put into words. I mean, I feel so grateful to work with such a strong team. If you look at our chief medical officer, he's he oversaw the only 2 approvals in lupus since the 1960s, and he brings with him an enormous amount of experience and expertise in his team. But then also I learned so much from our CEO, from Arun, in terms of thinking about what the product needs to be at the market at the very beginning, and then designing everything around that. We now have a chief commercial officer. I'm learning a huge amount in terms of what we need to do in order to make this commercially viable. And then, of course, the team members I work with, that I've worked with before and I'm working with again. It's an incredible experience. The one challenge with biotech usually is that you're waiting to see the signal in the clinic. You really believe the science, but you're waiting for that clinical signal. We have something that it is an enormous clinical signal, and it's all about execution and making every day coming in and understanding where the leading edge is and how we can make that critical decision just to stay at the leading edge, which is one of the reasons I think we as a small biotech company is perceived by many as leading the space from a clinical, translational science, even manufacturing perspective. And I think that takes a fully integrated team, a lot of messy, hard, continuous discussions, and because we've been together so long, we do that really well. And I just think that is a rare experience to have, and I'm super grateful to be part of it.
Yeah. I love that you described it that way, like having those messy, hard conversations, because they are so critical to be able to come to the best decision to move as quickly as you have been able to move. Like, I really do believe that's, even though I'm not in it, but having lived in bigger companies and smaller companies and seeing how teams operate, I do believe that like surfacing dissent and having lots of debate within your team really brings out the best solution to be able to then move as quickly as possible. So I love that you're saying that, and all of that, everything that you just talked about. I mean, it's homegrown here in Philadelphia. I imagine most of your team is from here. Like some, you went to Johns Hopkins for a little bit when you came back, and we love you that you did.
So did Gwen.
I did.
Yeah, so did Gwen.
That's right.
Yeah.
I did. Yes, I remember
Take
that.
all our best talent.
That's right.
Like you kind of leave, but then you come back because Philly's just amazing.
I love
And so
Philly.
It is.
I love Philly too.
Yeah.
So I'm so like, it's awesome to talk about like this story here. And I wanted to talk a little bit about like community engagement here in the ecosystem through your REAL initiative, which I have written as Reflection, Education, Action, and Leadership, a commitment to community engagement. So what does it mean to actually live like the values that are on your website? You're actually living them day to day. And is there a moment where you've seen the culture of Cabaletta here in the Philadelphia ecosystem really enable something scientifically or clinically that you don't think maybe would have happened otherwise, like as a result of the culture that you have built here? Can you think of a moment or a time that maybe it was really about the people and not just about the science?
So to me, and I'd be curious actually what your answer is here.
Yeah.
To me, it is the speed with which we took to evaluate no preconditioning in autoimmune disease with Rese-cel. And the reason is we were, our co-founders were co-founders who engaged in a collaboration at Penn. They were both Penn clinician scientists. And one of those co-founders, Mike Malone, was someone who was an inventor of Kymriah and was, as far as I know, published one of the only papers in the oncology setting that evaluated CAR T without preconditioning. BCMA CAR T in patients with multiple myeloma across a dose range that was published in 2019. It is a paper that I cannot tell you over the past 7 years how many times I have referenced. And sure, there's a scientific component of it, but the depth of relationship that Gwen has with Mike, that I have with Mike, that we both, that it's not just us, across the company, with Mike, with Amy, with others in the Penn ecosystem, there's so much scientific innovation that has come out right there that to be able to have access to it, we can walk over to their offices, go out to coffee, have a chat, like those sorts of connections bring you insight and know-how in a way that reading papers, doing whatever research you can do just doesn't deliver that same degree of richness. That for me is just one example. I'm sure it's come out in a bunch of different ways, but the comfort that we had with starting off with that strategy, and the reason I say that is it's taken other companies much, I would say, longer than I expected to come up, at least publicly, and no one has publicly come out with such a strategy, because I think they didn't necessarily have the confidence or the relationships, not the confidence, but the relationships to understand why might this work? Why is it different in oncology versus autoimmune disease? And these were questions that we've been asking since company formation. So I don't know if you have another.
Yeah, no, I mean, I think the strength with, having a scientific pedigree like Penn and many of us who've gone through there, I think is really huge. I'll take a slightly different tack, and it has to do with the culture of the company and the Get Real initiative and how that impacts our ability to execute, right? So I agree with what you said, and so the other thing is sort of the hard conversations you have to have in a company and the trust you have to have throughout all levels of the company in order to move quickly, especially in difficult times when finances are constrained or, there's a high levels of competition or we're changing strategies in order to keep up. That can erode trust and confidence within a company. And something that actually our former head of HR started was this Get Real program, and it was started I think around the time of the pandemic and also around the time of the George Floyd killing and all of the protests that were happening in Philadelphia and throughout the nation, and there's a huge amount of anxiety within the company, and we wanted to talk about it, and so she took this bold move to have those conversations within the company, and I think oftentimes there's a feeling that you should not have difficult conversations at a company because of liability or you might offend people, but through her help and the Get Real team, we were able to create pockets of safe spaces where we would have those difficult conversations. And I always felt uncomfortable in those. And then at the end of the conversations, I always felt amazed at how vulnerable and courageous everybody was. Yeah. And what that did is it allowed not only people to let off steam in a difficult time, which impacted each of us individually in our lives, but it built trust within the company. Right? And so now, there's other things going on in our society now that are really challenging. We can continue those difficult discussions. And I think what that does is it builds that trust so that when we ask, for a new project, there's trust throughout the company. And that's not why we did Get Real. Yeah. But I think that's a hidden benefit of the culture that leads to a lot of scientific and clinical progress. Yeah.
Gwen's answer is way better.
That's not
way better, way more on point with how, and it being comfortable. There's so many things that we can do to feel that can make us uncomfortable. Part of it and part of our ethos is everyone who comes into this company should be someone who is comfortable asking for, delivering, and responding constructively to feedback. And so if you think that what Gwen just referenced, all incredible examples of you've got to be uncomfortable to get to that next increment of progress. That is true with what I think is the fundamental thread through our organization, which is people show up to work ready to serve the team and ready to ask for how they individually can improve to better serve the team, what went well, what they can do going forward, and telling others, regardless of position, level, seniority, any of those things. It's if there's something that I can either say to someone or hear from someone that helps me do better for the team, I'm willing to do that. So I thought that was excellent.
I love it. Oh my gosh, I love that. And the thing that really struck me is their willingness to be uncomfortable. People are just prepared to be uncomfortable because that's really where you see the growth, the change, the innovation, and really people pushing boundaries. So that's amazing. Love hearing all of that. Okay, so I'm going to pivot a little bit more because I think this is a topic that doesn't get enough or doesn't go deep enough. We talk a lot about reimbursement mechanisms for CAR T therapy. And I just feel like nobody really talks about like the depth of what that actually means. And so, you know, we talked about inpatient therapy versus outpatient therapy. There's definitely differences there in terms of reimbursement. I love how you've been so transparent in your corporate deck. So thank you for giving me something to work off of, which has been really allowed this conversation to be as rich as it has, to be quite honest. And you talk about it. You talk about like the DRG reimbursement. You talk about Medicare Part B in it. So tell us a little bit, like, can you go a little bit deeper into like how you've been thinking about that, you know, the patient population that you're serving, how that all mixes into the strategy that you're taking and your preparation for as you mentioned, you just hired a chief commercial officer as you're preparing for potential launch, because you have the 2027 BLA potentially coming. So, you know, tell us more.
Yeah. So I totally understand why you're asking the question. Definitely something we can get into. I think it's helpful to understand, why are we asking this question in the first place? And the reason we're asking this question in the first place is for the CAR T launches in oncology that came before us, there were two big drivers that I think folks in the field would recognize limited the uptake of of therapy, not disease, limited the uptake of therapy. One is the payer mix within the treated population. And two is the setting of care that the CAR T was delivered. So in the oncology, let's baseline in oncology and then we can go to autoimmune. In oncology, the predominant payer for the treated patients was Medicare. In the autoimmune setting, the predominant the predominant payer group is commercial patients. In the oncology setting, the predominant setting of administration was inpatient administration based on the degree of illness in the patients and how severely sick those patients really were. That was the big driver. In the autoimmune setting, we are positioning, and here we'll talk about rese-cel in particular, but based on the safety profile of rese-cel across the phase 1-2 cohorts, we fully anticipate outpatient administration to be not only an option in the pivotal study for our first registrational study, but ideally the preferred the preferred administration in the commercial setting is outpatient. Now, the reason why these two things are somewhat interrelated is if you are a Medicare patient, and so let's go to the oncology setting for a second, and you are treated in the inpatient setting, which is where the majority of patients were treated initially, at least with CAR T, you are now subject to the DRG reimbursement system. That is a inpatient specific, Medicare specific system that guides reimbursement. And I think most would recognize that in the setting of reimbursement in that setting, it was perhaps not as optimal as folks would have wanted. And certainly with commercial payers, reimbursement is meaningfully different. So as we transition to the autoimmune setting, we see that the majority of patients are commercially insured. And from a setting of care perspective, the other reason why inpatient versus outpatient matters so much is resource utilization. When you're taking up a bed on a bone marrow transplant floor, and you take up the nursing time and the other positions that are supporting care in the inpatient setting, that's a significant drain on the system where it may be hard to prioritize an autoimmune patient over an oncology patient. Because we believe that the safety profile of rese-cel can facilitate it, if these patients can be treated predominantly on the outpatient setting, you've opened up a scale opportunity that can allow you at each individual site to treat many-fold more patients than you would if you were restricted on the inpatient setting. And so I think it's important to realize it's not just about reimbursement. Reimbursement's certainly one component of it, but it's also about resource utilization and being able to pair both the autoimmune payer mix and the setting of care is what we think gives us such a differentiated opportunity to make a commercial impact with rese-cel in the first few years of launch that is different than what we've seen with any other CAR T.
Yeah, thank you for kind of going into the depth of that, because I think people don't realize that it is two very different patient populations for so many different reasons, this being one of them, and taking that into consideration and how you're thinking about it ahead of you're really thinking with the end in mind of like, how are we really going to get this to patients and actually get paid for it and so forth. So, and that's going to be the best setting for them too. Like, it's kind of like if you will, like a really holistic approach. Just to kind of round out the conversation, Cabaletta was founded out at University of Pennsylvania in 2017. You're still very active in the ecosystem here. Philadelphia has such a richness of resources and people and talent. And so what do you believe makes Philadelphia genuinely competitive as a space for new founders to come and establish their headquarters here?
I can start. I mean, there's multiple things. So I've now started 2 biotech companies in the Philadelphia area over the last 20 years, or been involved in startups and built the teams. And from hiring people from different types of teams, scientific teams, clinical teams, manufacturing teams, having to find space for them, having academic collaborations. So Philadelphia is uniquely positioned in the nation to draw talent, from pharma talent from the suburbs, academic talent from all of the universities in the area. We are ideally situated between the financial center of New York and the regulatory center of Washington, D.C. We have an international airport that's easy to get to. The West Coast is easy to get to Europe. And real estate is relatively affordable in this space. And when you compare us to other biotech hubs, especially like Boston and so forth, and also because we aren't sort of such a highly strung competitive environment like Boston, once we do invest that time into talent and we have affordable space, we can have a better chance of retaining and building on that talent. So I'll hand it over to Arun to build on that. But I mean, I have time and again seen this as huge value to Philadelphia. There's also multiple spaces. There's Center City. There's Schuylkill Yards. There's Conshohocken. There's many areas around the city in which there are biotech hubs also that can fit the needs of any particular company.
Yeah. For me, it can be summarized in one word: accessibility.
Yeah.
So from where we sit here, I can see where I teach on Fridays, I can see where my family and I live, and I can walk between all 3 points. I can also, across, without even going outside, go to the train station that can get me to DC in an hour and 50 minutes, New York in an hour, and Boston in probably 4, although that one's probably not a ride you want to make if you can avoid it for too much. It's also about the airport. It's also about cost of living. I mean, it's honestly, I don't understand how Philadelphia has been able to, given its proximity to so many things, been able to grow and develop in a way that has maintained its accessibility. And that even goes down to how easy it is to walk from place to place. There's a reasonable public transportation system here. Like, it's incredible that all of those things have been able to come together, and I just feel like this city has been on and continues to rise in a way that is the place to go, especially in cell and gene therapy or in biotech innovation. It would be hard to find a better place.
Yeah. For cell, especially we're doing as the Silicon Valley.
Yes.
Especially for cell and gene therapy.
Yes.
Is there anything you'd like to see more support of in the Philadelphia ecosystem?
Yeah, I mean, I think one thing I'd like to see more of, and I know there's many groups working on that, is, I would like more accidental discussions with other people in our space. So like, if you're in Boston and you go out to a bar or a restaurant, you're running into other people who are like you that you can talk to. If you're in Washington, D.C., you go to a bar, you talk to other policymakers, they're just around, right? And so I think you need to put a little bit more energy to find those pockets within Philadelphia. So I would like to see a more active, opportunity for those types of interactions. They're there. People are working on it, but I would just like to see more.
Okay.
I think that's great. The opportunity for informal social gathering, and then specifically when we think about where are folks working in Philadelphia, a lot of it is in University City, and even in University City, there are places you can go, but there aren't sort of locale squares, areas where there's like a hub of activity,
Right.
where you can just go. Most of the places you go, you have to have a purpose. Like, I have a reservation here. I'm going with a group. This is why I'm going. I don't think that as much exists. Like, I'm just going to, after work, hop across to the square, to
what's
whatever
going
the
on.
square is, right, and see what's going on.
Yeah.
That I don't think happens as much. And that is I think it's great. I didn't think about it as much coming into this, but absolutely. I think that would be a big
the
improvement.
Square. I think that's a great way to solve the problem.
Yes.
I love it.
Interesting. Okay. More random interaction.
Yeah.
And sharing of ideas. So I want to close out with one last question about the reset program. So if it works the way that you believe that it can, it just delivers durable drug-free remissions for patients with all of the diseases that we talked about and maybe even more. What does the world actually look like for patients?
You know, one of our PIs said to us, you know, when I have a patient, I have to make a decision whether to, you know, give the patient a new disease by treating the disease that patient has. And I think for patients that have treatment refractory autoimmune disease, which is a significant portion of them, and most of today's treatments are not curable. So you have patients you have two types of patients. You have patients who can't be cured with current therapies, which are a majority, and then you have patients that even on therapy can't control their disease, which is another issue. But what the world looks like for patients with autoimmune disease is normal. You know, you're giving patients back the opportunity to live a normal life, take care of their kids, go to work, have energy, be able to focus, just to be themselves again. That's
I couldn't agree more. To me, it's, you know, I think maybe we're one of the first to come up with the idea of the phrase resetting your immune system with CAR T. This is really a chance for patients' lives to be reset if everything you said happens. Is that you bring them back to what they were like, or as close to what they were like as you can prior to disease onset, and all the things that you just talked about, Gwen, become realities rather than dreams. That would be a pretty great world.
Ugh.
Yeah.
I just got goosebumps. Well, thank you so much this was such an amazing conversation.
Thank
I
you.
really, really appreciate the time and the thought process and you sharing more of the behind the scenes that happens at the company. So I just want to do a quick Philadelphia lightning round, if that's okay.
Yeah.
So what's your best meal that you've ever had in Philadelphia and maybe does it involve a cheesesteak?
It involves steak. For me, it's steak frites at Park. So not a cheesesteak, but
good
steak
one.
frites at Park.
That's
That
a
is
good
awesome.
one.
Yeah.
So Park is a very favorite one.
Yeah, big favorite. Yeah,
That's definitely one of my
definite
favorite restaurants, I have to say, like sitting on the Rittenhouse Square. So not to go super fancy, but it is true. It is the best meal I've ever had in Philadelphia, and that's John George at the top of the Comcast Center. Can't believe they didn't get a Michelin star. Not sure what happened there,
Yeah.
but has been my favorite. And it's because it's such a great experience if you go with friends and to taste all the different things and to see all the different creations, and then the view of the city, of course, is amazing.
Oh yeah! Oh
Yeah.
my gosh! All those restaurants that are now at the top of the buildings are just
Yeah.
incredible,
They
incredible
are.
views.
Yes.
Incredible views.
Yeah.
How about a hidden gem in West Philadelphia that visitors never find?
There's that they never find. I'm not a huge West Philadelphia person, but I would say that, you know, I think visitors don't tend to go into West Philadelphia, but there's a giant park in West Philadelphia that always has vendors and performers and
Yeah, Clark Park?
Clark
That's
Park.
what I was going to say.
Oh, yeah.
Definitely Clark Park. Totally.
That's so funny
Yeah.
because when I lived there, it was not like that.
Oh.
So
We take
that
our
must
kids to
be-
Clark Park
That must be
on-
new.
Yes.
Or
Yeah, yeah.
newish.
At least to the farmers market, farmers market, but the stands, and there's a
Oh.
huge playground there, and on
That's
certain
so-
weekends they just line the
nice.
whole park
Oh,
with
that's
all these
so
pastries,
nice.
coffee, and we've absolutely our oldest has fencing, and he fences in West Philadelphia, and so we've taken the others into that area during his class or competition. It's great.
Oh, that
We
is
really like
so cool.
Yeah.
Oh, I love it. Yeah, that just brought me back to when I was at University of the Sciences.
Yeah,
That's
USP,
right
of course.
That's right on
Yeah.
our campus, yeah. So one word to describe Caballetta's culture.
One word.
I would say integrated.
Integrated.
Oh, one word is so hard. I want to come up with something with feedback, like feedback-oriented, feedback receptive, but I don't really like it, so I'll take what Gwen said. I think integrate. It is bringing your whole person to work, and everyone around the table recognizing that and being comfortable with it, embracing it, and that being critical to get the most out of the team. And I can't come up with a better word than integrated, so I think it's great. Yeah.
Awesome. The autoimmune disease that you most want to see Rese-cel conquer, and why?
It's just so tough. There's so many important autoimmune diseases.
It's not, to me, it's less about the disease and more about the approach. So I will say the thing I'm most excited about is to see whether we can go beyond the threshold dose of Rese-cel without preconditioning. And if that is able to bring about the same responses or similar responses that we've seen with preconditioning, to be able to eliminate that out of the paradigm of CAR T therapy and autoimmune disease would be, it's amazing as it is, even more so. That's me. It's less about the specific disease because I can't think of it.
Yeah, I figured out a way to cheat in my answer.
Go.
Yeah, my cheating answer is the one I want to see it most address is diseases caused by B cells.
Yeah. Perfect. Yes.
Sneaky.
Yes.
All right, last one. If you could go back and tell your 25-year-old self one thing about the career ahead of you, what would it be?
Don't get stuck on a plan, because you just have no idea what's going to happen. I stepped over from residency not ever thinking, even the year before, that I would do anything besides residency, fellowship, and become a clinician, at least for a period of time. And you just don't know what's ahead of you. And the more you can be open and pursue the things you're passionate about versus saying, I'm here at point A, I want to be here at point B, I think the better likelihood you have of being truly fulfilled.
Yeah, I think that's a really important message for people, especially today. It's impossible to predict the future. We thought it was possible back when at least I was younger, and but I approach it with an open mind. So I would tell my younger self, it's worth it. You're doing it the right way. Like don't worry.
Don't worry.
Don't worry.
Don't worry, and don't overthink.
Yeah.
Awesome. Well, thank you so much. This has been like such a dream and so much fun to have this conversation with you.
Thank you for the great questions.
Yes.
Oh my gosh.
This was wonderful.
And thank you for the amazing, just being open and going deep with some of the things that's really going on in the company. Like it's so awesome to hear behind the scenes. So again, thank you so much for your time.
Wonderful.
And look forward to seeing more of you, hopefully.
Likewise.
Definitely. Yeah. Thanks a lot, Joanna.
Thank you.